this is the last part of Chapter 2 in your book. So we're still on Chapter 2, but this is actually the 5th lecture and therefore the 5th quiz on organic molecules. So we still haven't really gotten that far as the organization of life. We've talked about atoms, why they interact with one another. We've talked about molecules, particularly water. Now let's talk about the most important molecules for life, and those are what the building blocks, or organic molecules as we call them. And most of us know the food version of these organic molecules that we consume on a daily basis. But there's so much more to it than that. It's not just about food. It's about structure. It's about function. So there's a lot of things you're going to learn new today besides just the fundamentals of what a carbohydrate is and what a lipid is and what a protein is and what nucleic acids are. Now before we get into that, there's one more property of water we haven't talked about. And I didn't include it in that other lecture, in Lecture 4, because it fits better with this one. And that is that in organic chemistry, which is essentially metabolism. Organic chemistry and metabolism are the same thing. It's just all the chemistry in cells in a living organism. And we've already talked about metabolism, the way that which cells and living organisms process energy. How they make energy, how they use energy. That's all organic chemistry is. The reason why we call it organic chemistry is because all of these molecules have at their foundation carbon. When we say something's carbon-based, that's what we mean by organic. Now the word organic today is used in different ways from the way in which we grow our food and the like. There's many definitions of organic, but the way in which we use it in biology, it literally means carbon-based molecules, because you'll see at the foundation of each of these groups, carbon, that atom, is at its center to build these biological molecules that are the building blocks for life. So where does water come into play? Because water is not an organic molecule, but it plays a key role in this. Water is involved in all metabolism. That's another property of water that you're going to be tested on in this lecture, your quiz 5, lecture 5. All of the metabolic processes of breaking down carbohydrates, assembling molecules into proteins and DNA and all these things, water is at its core. Without water, you don't undergo these reactions. So that'll be the first thing that we discuss today before we get into each of these 4 basic biological groups. Now, here are the 4 groups which we can categorize all organic molecules into, and most are familiar. Everybody's heard of carbohydrates. What do you think of when you think of carbohydrates? Breads, grains, sugars, things of that sort. But we'll show today that those are actually the minimum. The majority of organic molecules that exist on our planet are not for food. They're actually for structure. The plants and a great number of animals and other organisms use carbohydrates as their skeletal structures and their support structures for their cells, which really can't be used as a source of energy. So when we think of carbohydrates, we think food. When other organisms deal with carbohydrates, yeah, there's food, but they also use it for their overall structure. We don't. We don't necessarily use carbohydrates for structural components. We use other aspects of biological molecules. Lipids. When we talk about lipids, again, we know about triglycerides. You're going to learn the difference between saturated and unsaturated fats today on a chemical level. And you're going to learn about things like phospholipids, which aren't used for energy, but are critical for life, because all living things being made of cells will find that the major component of all cells' membranes are this organic molecule called phospholipids. Proteins. This is probably one of the most diverse groups. As such, we're only going to study the fundamentals of what proteins are and how they're made. But in the human body alone, there are about 400,000, just the human body alone, 400,000 different proteins. And that's not including other organisms. A lot of them have some of the same proteins that you and I have, but there's diversity within all life. And then, of course, the nucleic acids. We usually don't think of food when we think of nucleic acids. We don't have diets high in nucleic acids. So what are nucleic acids? Well, it's pretty much your genetic information. It's your DNA and what we call our RNA. But it also plays another role in energy transfer. There's a molecule in the nucleic acids group that is essential for metabolic energy transfer that we're going to talk about. So those are the four groups. So let's first start with water's role. These are two concepts that you're going to have two separate questions on: dehydration, synthesis, and hydrolysis. So let's talk about what they are and what's going on, and there's some more terminology you're going to have to learn. Dehydrate. What does to dehydrate something mean? Or when you're dehydrated? You're lacking water, and this is not hydrating me, by the way. In fact, they put so much salts and other things in the soda that you're actually dehydrating yourself when you drink soda. I still drink it, but you're not hydrating yourself when you drink soda with all of that sodium in it and everything else. Your body has to excrete more water than it actually can get out of the soda. So anyway, so when you're dehydrated, you essentially are losing water. Well, in that fashion, this process called dehydration synthesis is the organic chemistry of how all molecules are assembled together. So let's look at some terminology you're going to have to know. Each of the four basic biological groups has the building blocks which we call monomers. Mono, what do you think mono stands for? And I'm not talking about the disease. One. Okay, so monomers are single units. Now for each group they can vary. For carbohydrates, they're things like glucose. For lipids, they're things like glycerol and fatty acids. For proteins, they're what's called amino acids. You've probably heard of amino acids before. And in the nucleic acids group, they're called nucleotides. So the monomers vary in their overall chemical structure. But no matter what their overall chemical structure are, they are the building blocks for each of those groups. So when we build structural organic molecules like our proteins, like our carbohydrates, when we store it for long-term energy, triglycerides, which are the saturated and unsaturated fats, these are larger building blocks, and even your DNA. In fact, your DNA, the smallest DNA, is about 33 million monomers long. That's the smallest strand of DNA you have. So what happens? Well, when you have these individual building blocks that you typically get from your food, as you're eating your food, you're breaking down the proteins and the fats and the carbohydrates from other organisms, and you get this stockpile of monomers. Well, then we restructure them into the proteins and the carbohydrates that we need for our cells. So when monomers are assembled together, then they form long strings of monomers, which we no longer call monomers anymore. We call them polymers. So poly means many. So dehydration synthesis. Why is it called dehydration? Because as you can see here, in any reaction, whether it's proteins, nucleic acids, carbohydrates, or lipids, all of these reactions will remove a hydroxyl group and a hydrogen atom from the two monomers and remove water, and in that process will covalently bond the two monomers together. Remember I told you that a dash means a covalent bond. So this process is essentially building, taking smaller units and building them into larger units. So if we were to define dehydration synthesis, we would say that it assembles monomers into polymers. And let me say that again. So dehydration synthesis assembles monomers, covalently bonds them together, assembles monomers into polymers, longer chains that in some cases are hundreds of units long and others are millions of units long.
So the bottom one's a polymer.
This is a polymer right here. Yep. When you get these long strings of monomers, we call it a polymer. The top part are the individual monomers being assembled together. This is just done over and over and over and over again. This is also an energy storage process. When you build these together, you actually store energy. And we do this during periods of time when we're not eating. Our body will take some of the excess food and energy and assemble it together and then store it for later periods of time when we need that energy. Now, the opposite, called hydrolysis, hydro again stands for water, lysis means to break apart. So hydrolysis is just the reverse. It takes polymers breaks the covalent bond between the individual units by using water and essentially reverses this process. It breaks the covalent bond and it breaks them into their individual monomers. So, an example of this is every time we eat. When we eat starch, starch is a long polymer of individual glucose molecules. And when we eat that, our body will start breaking the individual covalent bonds using water and other things we'll talk about later in subsequent chapters, and break them down into their individual monomers, like glucose. That's one of the reasons why you get such a sugar high when you have what we call high fructose syrup, because the fructose is a pure monomer. There's no need to break it down. Your body just gets instant energy. And that's why you get the crash afterwards as well. So that's why starches and other large molecules tend to last longer, because your body slowly breaks them down and releases energy as you need it. You don't get this huge spike in energy where you start, going a little crazy. All right. So hydrolysis is polymers are broken down into monomers. This is what happens when we extract energy from these molecules. This is why we eat, to extract the energy from these molecules by breaking the covalent bond, and by doing so, we get energy out of them. So let's start with the one we're all pretty much familiar with, and that is carbohydrates. Carbohydrates, one of the main purposes of this group is energy. So don't get me wrong, even though I say that it's not the most abundant carbohydrates, it's all about energy. Plants, animals, fungi, bacteria, they love carbohydrates. It's the easiest organic molecule to break down and get energy from. That's why we go for the carbs first, is because our cells and the way that organic chemistry works, it's really easy to get energy from them. Easier than getting energy from fats. When you exercise, if you do that thing, you burn your carbohydrates first, and then your body starts digging into the fat reserves, which is why you have to typically work out for longer periods of time than just quick bursts, because your body will say, well, I got this store of carbohydrates, I'm going to go for that first. Okay, now, the name actually tells you what they're made of. Carbo has to do with carbon. Hydrates, like water, what's water made of? Hydrogen and oxygen. So guess what? Carbohydrates are made of carbon, hydrogen, and oxygen. That's their fundamental structure. So as they get assembled together, let's look at their fundamental building blocks, which we call, instead of saying monomers now, we have specific names for each group. These monomers for the carbohydrates, these simple sugars, we call them monosaccharides. So when we say monosaccharides, I don't have to tell you which group I'm talking about. You know that they're the monomers of the carbohydrate group. So monosaccharides, what are some examples of these? They can vary in their structure and shape, but they pretty much have the same purpose. They're pretty much used for energy. There's no other real purpose for monosaccharides. They're just energy. When they start getting built into larger structures, then they take on different functions. But for, in their monosaccharide form, it's energy. The three on the right. These are the building blocks for the carbohydrates. Glucose is by far one of the more prominent ones that's used in biology. But there's others, like fructose. That's what's in most of your candy and other sugar drinks and whatnot. High fructose corn syrup. That's it. It's just pure fructose monosaccharides that are being pumped into your body. And then there's one little less known called galactose. So glucose, fructose, and galactose. These are three types of monosaccharides. You will not need to know their structure and their shape and other things like that. You just need to know those three names, okay? And that they are monosaccharides. Glucose, fructose, and galactose, okay? Now, those are the building blocks. So let's start building. When you start undergoing what we call dehydration synthesis, which we just talked about, in the carbohydrate groups, you take a hydroxyl from one sugar, a hydrogen from another, and you covalently bond them together. So they end up having this oxygen between them, but the covalent bond binds them together. Well, now that there's two sugars, we call it a disaccharide. Di means two. Now, this is the type of sugar that you typically deal with when you bake. You know, we usually don't use high fructose corn syrup when we bake. What do we use? We use sucrose. That's table sugar. That's what you buy at the store in those big bags, which we call sugar. It's actually sucrose or a disaccharide. Now again, this is also used for energy. You know, we put it in to make things sweet. Our body can and does break it down through hydrolysis into glucose and fructose. So it's a very simple sugar as well, but it's not a monosaccharide. We call it a disaccharide. There's two other types of disaccharides that you're probably familiar with that I'm not even going to ask you for sucrose what the two monosaccharides are. But let's talk about some examples of some other disaccharides. Lactose, what's that found in? Milk. Okay, so lactose sugar is also a disaccharide made from two monosaccharides. And then there's one more. Maltose. Anybody venture a guess where you might have that? Or what it's used for typically? Like a malt liquor. It's usually the sugar used in a fermentation process. They use the malt in the malting process for fermentation. There's different types of disaccharides and sugars. Let's talk real quickly about lactose. I'm lactose intolerant, severely, and most people are. In fact, if you're not lactose intolerant, if you can break down the lactose sugars in milk, then you are a mutant. Not a very powerful mutant, but you are a mutant. I don't think you would join the X-Men anytime soon, but the reason why you're a mutant is because the normal process in humans is we stop producing a protein called an enzyme later on when we reach adulthood, where we no longer need those lactose sugars from the milk. Now there's other things that you can get from milk, but most of the time we replace milk with beer or something else. So we don't need milk. We need it when we're young, and our body produces that enzyme, but as we get older, we stop producing that. But evolution, we'll talk about, there, especially here in the U.S., has selected for individuals that do, because of our diet, have the ability to retain that the function of that enzyme and therefore break it down. Now, if you really want to have your ice cream and milk without the side effects, you can go down and buy enzymes. They're called Lactaid from Walmart or any other Walgreens or whatnot, and they temporarily give you that ability to break down the lactose sugars through hydrolysis. But we'll learn in lecture 7, when we talk about enzymes, how if your body doesn't make an enzyme, you don't undergo that process. Specifically, hydrolysis or dehydration synthesis. When you lack those enzymes, there's anything from a mild irritation in breaking down milk sugars to death if it's an enzyme that's absolutely critical for the function of certain organic processes. And this is where inherited genetic diseases typically come about, is when we're missing that protein due to the genetics we receive from our parents, it can actually be very life-threatening. Now, lactose intolerance is not life-threatening, but others can be. Okay, so sucrose, lactose, and maltose. You just need to know them as disaccharides. You don't need to know which ones make them up. You know, that glucose and fructose make sucrose. I can't even remember which ones make lactose and maltose. I know galactose is in here somewhere, but so just know those as disaccharides. They're also only used for energy. There's no other purpose for these disaccharides but energy. All right, now let's talk about the more complex structures of carbohydrates, what we call polysaccharides. Again, these are the polymers. So anything typically over about 100 monomers we call a polysaccharide. I make that distinction right now because at the very end of this portion I'll show you one more type of saccharide that doesn't really fit into what we're describing here. So polysaccharides, there are 4 that you're going to learn about and that you'll need to know for testing purposes. Well, 3 out of the 4 are up there. So we have cellulose, chitin, that's the one that's not up there, starch, which everybody's heard of, and glycogen, which not everyone necessarily knows what it is. All right, let's start with the ones that we're familiar with or somewhat familiar with. Starch and glycogen. These are almost identical. As you can see, each one of these green hexagons is a monosaccharide. So these are long, long strings of these monosaccharides attached through dehydration synthesis to form these polysaccharides. Okay? Starch is how plants store their carbohydrates. So that's why plant material typically is high in starch. Corn, rice, wheat, and all these things have these starch. Potatoes and the like have starches in them that we use for eating and whatnot. But we don't store it. Animals don't store their polysaccharides as starch. When we eat the starch, we break it up into individual little monosaccharides and we get energy out of it. And whatever's left over, we restructure it through dehydration synthesis into a slightly different molecule. It's a little more highly branched, as you can see, called glycogen. We put that in our liver. Now, why do we do that? Because in order to maintain homeostasis in our blood, we need to have reserves of sugars. But we don't reserve them as the individual monomers. We reserve them as these longer energy storage molecules, which we call polysaccharides. For animals, we store it as glycogen. But the same thing happens. When our blood sugar starts getting low, in order to maintain homeostasis, we start breaking off the monomers through hydrolysis by taking water and breaking the covalent bond, and then we reestablish that blood sugar level. Okay? Our body is constantly spending time to make sure that we have enough and adequate sugar in our blood so that it gets to our brain and our cells and gets to the rest of our body. So those are two examples of long-term energy storage for carbohydrates. Okay? Now let's talk about the other two. Notice the structural difference of cellulose. So where is cellulose found? Well, plants not only structure their monosaccharides into energy storage, but they also use it for structural support of their cells. What am I talking about? Wood, fiber, that's cellulose. So cellulose is a protective carbohydrate, very thick, very tough layer. I mean, wood is very tough because of all of these fibers of cellulose. Now, this is not an energy molecule, not even for plants. Why? Well, it gets into the fact that these are linear rather than branched. Again, in Lecture 7, when we talk about enzymes, we'll show that due to the fact that this is branched, we can break it down through dehydration synthesis. But due to the fact that cellulose is linear, we can't. Yeah, the energy's there. That's kind of the holy grail of trying to take plant material and actually extract it and make ethanol and things of that sort, which is a difficult process to do. But the energy's there, and some organisms can get energy from it. For example, termites. In fact, it's not even the termites that get the energy from it. There's a little microbe inside their gut that can convert the cellulose into monosaccharides, and then the termite gets energy from that. So, cellulose. Let's look at the plant fibers. They're held together due to their polarity by hydrogen bonding. Here's another example of how hydrogen bonding plays its role biologically. Just like water has its attraction to itself due to hydrogen bonding, so do the fibers or strands of cellulose have that weak attraction. Now collectively it becomes very strong. Individually the hydrogen bonds are weak, but collectively they're strong. So like celery. You eat a lot of celery, that has a lot of fiber in it. Not a lot of calories, not a lot of energy you can actually get because most of the celery is water and cellulose. So fiber is good for your diet, but you don't actually get any energy out of it. Now other things can highly compact the cellulose, and that's where wood comes into play. That's where trees get their very rigid structure, is due to these massive strands of interwoven cellulose fibers that form around their cells and give their cells a rigidity that you and I just don't do. All right. Now, so cellulose is by far one of the most abundant polysaccharides on Earth because it's the primary component of plants. It's the primary structure that surrounds the plant cells and gives them their rigidity. Now, chitin. Everybody wants to say chitin when they see it. It's chitin. Chitin is a carbohydrate that is primarily found as the exoskeleton of crustaceans and insects, crabs, lobsters, and the insect clade. This is their, when you step on something and you hear that crunch, that's chitin. When you go to Red Lobster and you crack open their shell, that's chitin. You don't get any energy from chitin. If you've ever tried chewing it, you don't get any energy. You want the meat, the protein that's inside of their legs or their body or whatnot. So that hard shell, that exoskeleton, is pretty much what protects these animals from, it's their skeleton, just like we have an internal skeleton, they have an external skeleton, but it's not made of calcium, it's made of chitin, which is a carbohydrate. So that's just another example. Now, chitin can vary in its overall form, which is why you're not seeing its structure. But it's still undigestible in terms of getting energy from it. So cellulose and chitin are what we call structural polysaccharides. They're not used for energy. They're used for structure, for plants, for animals that have an exoskeleton. It's used in fungi. Fungi do the same thing that plants. They surround their cells with carbohydrate, but they don't do it as cellulose, they do it as chitin. That's why fungi are so resilient in their environments and so difficult to get rid of, is because they can withstand extreme environments. I mean, they live in extreme environments. They're constantly degrading things and waste products. It's not an easy place to get food, so they need to protect themselves by covering their cells with this hard substance called chitin. All right, so those are the main groups in the carbohydrate groups. Now there's one more. This one doesn't really fit the mold, but you still need to know about it because throughout the semester you're going to be seeing this when we talk about blood type, when we talk about organelles of the cell, like the Golgi apparatus and whatnot. So it's going to come up several times, so it's important that we understand it. It's not a polysaccharide because it's not hundreds of units long. It's not a mono or disaccharide because it's more than 2. So if it's somewhere in between about 3 to 100, we call it an oligosaccharide. So what is an oligosaccharide? Because it's not used for structure, it's not used for energy. So what is it used for? Well, we know that if you need blood, let's say you have blood loss or you're donating blood because, or whatnot, that blood types need to match. A with A, B with B, AB with AB, and O, which is actually a universal donor. We'll worry about the RH factor later on, the plus or the minus. But the O, so we know that there's compatibility and incompatibility between our cells. And it's not just our blood, it's our organs. You do a heart transplant, you need to have a good donor match. What is it that they're trying to match? Well, on the surface of all of your cells, we have these, what we call oligosaccharides, which are essentially just 3 to 100 carbohydrate monomers that are covalently bonded to each other. So what is the purpose? The purpose is cell recognition. How do we know when our bodies get infected with a bacteria or fungus? It's because bacteria and fungus have their own oligosaccharides. And when our immune system sees those, says, oh, that's not me, and it tries to destroy it. So as our immune system develops, we learn to ignore our own oligosaccharides and destroy anything else. And that's why when they try to find a good match, a donor for an organ or for blood or whatnot, they need to have almost identical oligosaccharides on that tissue. Otherwise, it's going to be rejected by the body's immune system. So these oligosaccharides are attached to the surface of all your cells. For your blood, that's how we determine blood type. If you have one type of oligosaccharide on your blood, you might be blood type A. If you have a different oligosaccharide on your blood, you're blood type B. If you have none of the ones we usually consider for blood type, that's what O is. O is actually lacking these. And that's why O can be given to anybody, minus the other factors, the RH factor, the plus or the minus, because there's nothing to recognize as foreign. That's why O blood type is the most desirable, is because it's lacking these recognition molecules that will be rejected if the body says, hey, this isn't my blood, this is some foreign object. So that's what oligosaccharides are. They're in all your cells, they're attached to the surface of all your cells. Each person has kind of a unique set of these. The more genetically related you are, the more likely you are to have these same structural oligosaccharides, which is why they, when they look for donors, they first look for genetic relatives, because it's more likely to occur within people who are genetically similar to you. Lipids. This one's not as clear-cut as the carbohydrates, because there's a variety of groups within the lipid group. It's not like carbohydrates where there's 3 monomers that pretty much build everything. Lipids, there's a lot of diversity. The unifying concept of all lipids is that they are either completely or at least mostly hydrophobic. What does hydrophobic mean again?
Repel water.
They repel water. They don't like water. And the main reason for that is because they don't form polar covalent bonds and are therefore neutral. And water doesn't like neutral substances. It likes polar, it likes ionic. So water and oils and lipids don't mix, primarily because lipids don't have any charge to them and therefore will separate out from water. That's why they're hydrophobic. All right, so we know about a lot of the lipids that we have in our diet. But let's look at what makes the difference between saturated and unsaturated fats. Those are what we usually think of when we think of lipids as far as a food source. But there are other structures as well that become important. So there are 4 main groups in this, 2 of which I'm going to primarily focus on: the triglycerides and the phospholipids. We'll mention sterols and waxes. There may be one question on sterols, but the majority of the questions in this group are on these 2 because these play the most relevant role in all living organisms. So the triglycerides and the phospholipids. Now, yes, we do have monomers and polymers in this group, but they're not like the other groups where you have millions of monomers put together to form polymers, or even hundreds of monomers put together to form polymers. So technically speaking, dehydration, synthesis, and hydrolysis still apply to this group, but you're not going to see the large structures that are found in the other 4 groups, or the 3 of the other 4 groups: the carbohydrates, the proteins, and the nucleic acids. Okay, so as I mentioned, there's a lot of diversity within this group. Now, when we think lipids as a food source, we usually think triglycerides. Triglycerides are the saturated and the unsaturated fats in our diet. So let's look at the structure of it and then look at what saturated and unsaturated actually mean. So remember I told you how glycerol is technically a sugar, but it's only found in the structural component of a triglyceride. That's why we don't consider it a monosaccharide in the carbohydrate group, because it belongs in the lipid group. That's what triglycerides are made of. So the glycerol is 3 carbons with some hydrogen and oxygen. But really what makes it a fat are these huge long tails of carbon and hydrogen, which we call fatty acid tails. Now there's 3 of them, hence triglyceride. You have the glycerol and 3 fatty acids attached to it, and that's why we call it a triglyceride. All triglycerides have the same fundamental structure. Some can be a little bit longer. Some have a little bit longer chains than others. They don't always have the same exact length. But notice that they're very simple. They're just carbon and hydrogen chains. Remember, carbon and hydrogens share covalent bonds equally. And that's why this molecule has no polarity to it, has no charge to it. It is absolutely neutral, and that's why water and oil do not mix. All right. Now, what does saturated and unsaturated fats mean? Well, it has to do with the covalent bonding of the fatty acid tails. If all of the carbons are single covalently bonded to each other, and they're saturated with the hydrogens, that's what we call a saturated fat, is when the tails of the triglyceride have the maximum number of hydrogens attached to the carbons. Okay? But occasionally you'll get fats that have double covalent bonds in them. Well, what that does is, because these two carbons are sharing 4 electrons instead of just 2, that reduces the number of bonds it can form with other atoms. Hence, you get these gaps where there's no hydrogen bound to the carbons, and so they're unsaturated. Now, we have monounsaturated fats, we have polyunsaturated fats. That just tells you how many double covalent bonds there are. If there's one, it's a monounsaturated fat. If there's many, it's a polyunsaturated fat. So, structurally, what's the difference between these two, and where do we find them? Well, here's just kind of a bubble filling model of a saturated fat. Notice all of the tails are perfectly linear. They're all saturated with hydrogens, and it's very compact. These are what we would consider beef fats, the more solid fats, lard and the like, that really aren't as good for you health-wise, because though they pack a punch, they've got a lot of energy to them, they're harder to digest than the unsaturated fats. So saturated tend to circulate through our cardiovascular system more often. They have a higher chance of being deposited in our arteries and causing clogs and things of that sort. But pound for pound, fats have twice the amount of energy as carbohydrates. I mean, look how many covalent bonds there are here. And every covalent bond gives you potential energy. So that's why, though, if you have a pound of sugar and a pound of fat, you're going to get twice the amount of chemical energy out of the pound of fat, because of how condensed it is and how many of those you can have. All right, so what does an unsaturated fat, especially a polyunsaturated fat, look like? It looks like this. So because of the double covalent bonds, the tails are no longer linear. They're just kind of all over the place. Well, that creates they're less dense and therefore more fluid. So unsaturated fats are things like oils. They're much more fluid and not as compact. Now, these are much easier to break down and don't spend as much time in our cardiovascular system, which is why these tend to be more good for you. Now, you're not lubing yourself up on the inside with oil, so that's not how it works. But you want to think about it that way. That they're better for you. Now, can we turn a polyunsaturated fat into a saturated? Absolutely. That's what margarine is. Margarine is where they hydrogenate the triglyceride, meaning they break the double covalent bonds and they add hydrogens to it, thus turning it into a saturated fat. So what is margarine? It's a solid oil. That's why I don't touch the stuff. It's just nasty. So margarine, there's pros and cons to all things. Due to the chemical process where they create these trans fats, it makes it very difficult for the body to break them down. So anyway, we won't get into all the health reasons or whatnot. But as far as fats go, the saturated fats are things like the cheeses and the ice cream and the lard and the butter and whatnot, where when you get to the oils that you get from seeds and other parts of plants and other tissues and whatnot, those are the unsaturated fats. So that's really the main difference between the two. It comes down to the overall number of covalent bonds between the carbons and the hydrogens. And the more double covalent bonds there are, the more fluid the triglyceride is, and those are where you get the oils. Okay. Now, that's our food source. Now notice, they're put together in the same way that the other molecules we've talked about are put together. The individual monomers are the fatty acids and the glycerol. When you remove water from each of these bonds, that's dehydration synthesis, you covalently bond them together and you make a triglyceride. So in that fashion, you still have dehydration synthesis. So what's hydrolysis? Well, we do the opposite. We come in here, we break these covalent bonds. So when you get triglycerides in your diet, first you break off all the fatty acid tails. Then your body comes in here and starts chopping off these covalent bonds through hydrolysis, and those two carbons actually pack quite a wallop. You get quite a bit of energy from that, which is why fats give you more energy than carbohydrates. And that's why your body, it's a little harder to get at, which is why it says, oh, I'm going to keep that in reserve. So triglycerides are good for long energy storage. And yes, you do form triglycerides if you have too much sugar in your body. And it all depends upon the individual's genetics and their metabolism on how fast they convert excess sugars into not only glycogen, but they can reconvert the carbohydrates into fatty acids. And yes, you can increase your fat tissue by just eating carbohydrates. You don't have to say, well, this is low in fat. So anyway, there's always trends and other things, and I just try to dispel some of these myths. All right. Phospholipids are almost identical in their overall structure to triglycerides. Let's break it down. It has a glycerol, just like a triglyceride. It has fatty acid tails, and those tails can be saturated or unsaturated, just like triglycerides. But instead of having one more fatty acid tail, which is why we don't call it a triglyceride, attached to that third carbon is a phosphate and a nitrogen group. Now, this is what makes this molecule not only unique, but essential for life, is that group, that phosphate and that nitrogen group. The phosphate group is negatively charged and the nitrogen group is positively charged, which creates a polarity to this head. And we call this portion of the phospholipid the phosphate head or the hydrophilic head. When something is charged, it likes water. So this molecule has a personality crisis, so to speak, because the majority of it hates water. These hydrophobic tails, being neutrally charged, don't like water one bit. But this head loves water. So what happens is when you put phospholipids into a watery environment, remember all living things are mostly water, it does this. It creates these bilayers, two layers of phospholipids, where the heads are oriented on the out and inside of the cell, and the tails are oriented on the inside where they're not near water. This creates a fat layer that protects the cell. We call this the cell membrane, and all cells have it, which is why this is one of the most crucial molecules for life, is because all cells are surrounded by these phospholipids bilayers, as we call them. And that's what helps separate the external and internal environments of the cell, where it's able to maintain homeostasis. Every living organism has these. This is not used for energy. It is purely for structure. All cells have this, not only on the outside, but as we'll learn in the next lecture, on the inside as well. These phospholipids form the majority of what we call organelles, or the organs of the cell that each have specific functions. The majority of them have at their foundation these phospholipids. So it forms a very important structural component for the outside of the cell and the inside of the cell. Now, less talked about, but still important to look at, this is where things start getting a little crazy. Because unlike triglycerides and phospholipids, sterols are totally unique. They do not have even close to the resemblance of what triglycerides and phospholipids have their fundamental structure. And these really don't form polymers. This group, the sterols and the waxes, don't really form huge polymers or even close to the polymers like triglycerides and phospholipids. So what are sterols? We usually think, when people think sterols, they think steroids, and those are actually different. So don't confuse the two. Sterols and steroids are different. So what are sterols? Well, they're fats because they fit the definition that they are hydrophobic. But notice their structure. It has no resemblance to the triglycerides. So what does it look like? Well, the main fundamental structure of all sterols is what we call a 4-fused hydrocarbon ring. Okay, just remember that word. 4-fused hydrocarbon ring. It's not even like a sugar. Sugars have these big gaps in oxygen in between them and form these long. These are actually fused together. That's why we say 4-fused hydrocarbon rings. What is the most prominent sterol? Cholesterol. So cholesterol is not a triglyceride, but it is a fat. And our body uses it for a number of things. It uses it and puts it in our cell membranes to increase their fluidity. We put it in other parts of the cell as a hormone, so to speak. And we convert it into various hormones. Cholesterol gets turned into testosterone and estrogen, which are also sterols. So testosterone, we know about. Estrogen, we know about. By the way, the myth that there's more estrogen or testosterone in male or female, let me just take that away. Men, in your testes, you have a higher concentration of estrogen, which is used to create your sperm, than women have circulating through their body. So we use both, women use both, testosterone and estrogen. Yes, there are differences in where we use them and how we use them, but ultimately testosterone and estrogen are both in male and female. Just depends upon where you look. Vitamin D, cortisone. Cortisone is an important hormonal signal. And vitamin D plays a key role in from our skin being activated by light and then creating the vitamin D that we need for various aspects of our body. But not all organisms typically use sterols. You don't get bacteria using testosterone. You don't get other microorganisms using a lot of these. So that's why it's not necessarily talked about as much. But it is important to at least understand that cholesterol is not a triglyceride. It's a sterol, as we call it. Now, I have got some beehives, so I love talking about this. I forgot to bring in my comb. Usually when I do, people start ruining it and they squish it and whatnot. I'll bring it in next time. But the waxes are also, again, very unique. Now, they are lipids, but they're somewhere in between a saturated and unsaturated fat. And they're not really triglycerides. They do have fatty acid tails, but they are combined with alcohol or other carbon structures. So what's so important about waxes? Well, they are hydrophobic, and they're great for, like in the bees, for storing the liquid honey. We use wax in our ear to be able to collect and prevent organisms and such, help with dust and other things that's necessary. But not all organisms use waxes. So a wax, that's why you're not even seeing the structure here, is a water-repellent, pliable substance. So it is hydrophobic, and it's used in very unique circumstances, but not all organisms necessarily use them. That's why this one's kind of an oddball as well. The proteins group is the most diverse of the 4 basic biological groups. In the human body alone, you have 400,000 different proteins. That's a lot. And so ultimately the question becomes, what are the fundamentals of the protein group? Since we can't go over all 400,000, what makes a protein a protein? What are the fundamentals? All right, so let's start with the basic building blocks. Monomers. What are the monomers of the protein group? Well, we call them amino acids. Now, you've heard of amino acids before. Typically, you hear about them in nutrition, where you talk about essential amino acids and non-essential amino acids. Let me tell you the difference between them. Essential amino acids are amino acids that you can't make yourself. So you have to get them in your diet. You cannot manufacture them through metabolism. So you have to get them in the food that which you eat. Non-essential amino acids are ones that you can make from other molecules that you consume on a daily basis. So that's really the difference between them. You have your essential ones that you need to get through your diet, and then your non-essential ones which you can manufacture in your own cells. So what is an amino acid? Well, the name, it gets its name because of the chemical groups that are universal for all amino acids. On one end, you have a nitrogen and two hydrogens, which we call an amine group. And then you have a carbon, which is attached to in the center. And then on the other end of that carbon is what we call a carboxyl group. Now, without going into the details of how this is done, the carboxyl group actually releases a hydrogen ion, which makes it an acid, hence amino acid. That's why they're called amino acids, because all monomers of the protein group, all amino acids have this fundamental structure. Now, what's this R group right here? That's what makes amino acids different. There are 20 amino acids that all living things use as their building blocks. They are the universal building blocks for all proteins. In some situations, it might be a simple hydrogen atom. That's one of the most simple amino acids called glycine. In other situations, they might be huge carbon ring structures with some nitrogen in them as well, like tryptophan. Here's another one called cysteine. You're not going to have to memorize any of the amino acids, but these are just some examples of how amino acids can vary substantially. And there are 20 of these building blocks. So let's look at how they're put together. No matter what we call R, or functional group, is, they all have the same basic fundamentals of how they're assembled together. Same process as we talked about before. Hydrogen and hydroxyl group get removed through dehydration synthesis, and the covalent bond exists. Now, the book and others will refer to this as a peptide bond, but it is not a new bond that you haven't learned. It's a covalent bond. Why do they call it a peptide bond? Because chemically, chemists, because of the nature of this bond, call it a peptide bond. So the reason why I point that out is because it makes it easy to understand what the polymers of all proteins are. They're called polypeptide chains. That's the universal word for all proteins. All proteins, when they're assembled together from their monomers into a polymer, we call them a polypeptide chain. Doesn't matter which protein you're talking about, all proteins are polypeptide chains. Essentially just long strings of amino acids assembled together through dehydration synthesis. Okay? So, make sure you understand, peptide bond is no different than a covalent bond. It's just how chemists name it. Now, the same process occurs when we eat proteins for a food source. We break them down through hydrolysis. We add water, we break the covalent bond, we get energy out of it. So we can get energy out of proteins and carbohydrates and fats. Well, what's interesting about proteins is not that they're not just for food, they're not just for energy, but they're what make you who you are. Let's look at the composition of what actually is a protein. Proteins are not just simple linear structures. There is a three-dimensional shape in which they fold into that gives them a very unique role in the cell or in your cells. Let me give you an example. We know that wrenches and screwdrivers are tools that are used for various jobs. They're pretty much made of the same material. Fundamentally, they're made of metal. But based upon what mold they were put into when that metal was shaped will determine what job they can do. You can't use a wrench for what a screwdriver would do, and you can't use a screwdriver for what a wrench would do. And the same thing applies to proteins here. Proteins essentially have this hierarchy of folding that is based upon their ability to covalently bond, hydrogen bond, and here's where we get some ionic bonding. Yes, there is some ionic bonding in the protein structure. Remember we talked about ionic bonds typically don't form, but this is the one exception where in some scenarios when the protein folds there is some ionic interactions between the protein chains. So let's look at this hierarchy because this is what I'm going to test you on. The primary structure of a protein is essentially the order of the amino acids. Now, the order is just what order do they get covalently bonded in? Alanine, threonine, cysteine, tyrosine, glutamate, glycine, and whatnot. What order do they get put in? Well, just like you understand how in the English language, the different order that the letters are put in have different meaning for the words, The same thing is true for proteins. What order the amino acids go in are covalently bonded to each other predetermine how that protein is going to fold and therefore function. If you put them in the correct sequence, then they will fold properly into that globular or three-dimensional shape, like a wrench or a screwdriver, so to speak, as I gave you that analogy. But all the primary structure is the order of the amino acids, nothing else. How do we get that order? Well, this is the relationship between your genetic material and your proteins. Your DNA, which is found in all of your cells, has that template that tells your cell what order to put the amino acids in and therefore how to make your proteins. So in lecture 10, still a ways off, but lecture 10 is when we're going to discuss very deeply this relationship between how you go from the blueprint to the actual house. It's kind of the difference between having a blueprint of a house and the actual house itself. The DNA would be the blueprint, the house would be the protein. That's how you construct it as your blueprint tells you how to do so. Now, the secondary structure is where you start to get initial folding. It's not quite ready at this point, but you do get some of the, think about origami. In the initial stages, you're just kind of folding it. It does have some dimension to it, but it's not what you want it to be yet. It's just the initial stages of it. So you start getting these loops and these little sheets and whatnot, but proteins really don't function on that level. Now, where does the secondary structure come from? It primarily comes from the hydrogen bonding, because amino acids, like other molecules, have polarity to them. Now, there are some amino acids that are neutrally charged, but there are a lot of other amino acids that have a polarity to them. And then there are some amino acids that are ions. So there's a great amount of variety of amino acids. Some are neutral, some are positive charged, some are negative charged, some are polar, which they have both charges. There's a great deal of dynamics. So you can see here that these loops and sheets actually form because of the polarity of these amino acids. They're like water and like carbohydrates and like and whatnot, as I showed you. This is another example of hydrogen bonding as it exists in biology. Well, once you get through the initial secondary stage or secondary structure of folding, then we get to the most important one, which is what we call the tertiary structure. So what's the tertiary structure? It's essentially the three-dimensional shape that the protein will take on, and that gives it its function. This is the difference between the wrench and the screwdriver and the hammer. They each have respective jobs, even though they're made of the same material. And one can't do the job of another. Okay? So tertiary, you'll see on the quiz times the use of the language three-dimensional or globular. Those all mean the same thing. So tertiary, globular, three-dimensional, these are all, space-filling models of how the protein is structured. This is pretty much how all proteins function on this level, is this tertiary structure. And we'll give some examples of these. Now, the last one isn't actually much different than this one. It's called quaternary structure. What is quaternary structure? It's essentially multiple polypeptide chains all combined together into an even larger three-dimensional structure. So the only difference between a tertiary and quaternary structure is how many polypeptide chains you have. For a single tertiary structure, it's just one polypeptide chain folded. But for a quaternary structure, a perfect example of this is hemoglobin, which is in your blood. Hemoglobin actually has 4 different polypeptide chains, each individually folded, and then what holds them together in the middle? Iron. That's why you have a lot of iron in your blood. So there's some iron that holds these 4 together. That's what carries your oxygen and your carbon dioxide through your blood. Okay? So the only difference between these two is just how many proteins are involved, how many polypeptide chains. So if you see me describe multiple polypeptide chains forming a larger structure, that's quaternary. But as you can see, they're really not much different than each other. In fact, this is generally the rule, this is generally the exception. There are more proteins that form these higher complex structures in your body than single proteins. Now, this is just to give you an example of some of the proteins. You're not going to have to memorize any of these, but some of these will come up quite a bit throughout the semester. So it's good and important to at least give you a preview about what we're doing. Muscle. Your muscle cells have two major proteins. There's actually more than two, but two major proteins, actin and myosin, which interlink and ultimately are the proteins that cause the force of contraction. So your muscles, which are cells, are able to function because they have an abundance of these proteins. Antibodies. Your body produces 10 million different variations of antibodies to try to respond to any infectious disease. Carbohydrates, lipases, proteinases. This is a huge group that we're going to spend a lot of time on lecture 7, called enzymes. Guess what's actually doing the dehydration synthesis and the hydrolysis? It's enzymes. So they're proteins that are actually putting molecules together and breaking them down. So it's not just happening spontaneously in your cells. These enzymes are what actually break it down. If you remember, I talked about lactose intolerance. People who are lactose intolerant can't break down the lactose sugars because they fail to produce this enzyme that does that. And no two enzymes do the same job. Each one has their own tertiary structure and therefore does only one job. It's like trying to take a wrench to do a screwdriver's job. It won't work. Okay? So each protein has a specific job. If you don't make that protein, you don't do that job. In some cases, it just causes mild irritation of milk proteins. In other cases, it can be life-threatening. So, let's see. Insulin. Insulin is a protein we're going to talk about quite a bit because it's necessary for your body to maintain homeostasis and pull glucose out of the bloodstream into your cells. We know if you have type 1 diabetes, your body doesn't produce insulin, and so you have to have insulin injections to regulate your blood glucose levels. Type 2 diabetes is your body doesn't respond to insulin, and there are other mechanisms such as exercise that can actually help mitigate the absorbance of glucose into your bloodstream. Keratin. Keratin is one of the more diverse proteins because it's found in so many different tissues in your body, and it can be compacted in any number of different ways. It forms your hair, it's in your skin, it's in your nails, it's in the horns and beaks of various other organisms. So it can be really, really tough. It can be slightly tough. It can be very pliable, like in your skin. It can be tough, like in your nails. There's many other things, but keratin has pretty much one main job. It's a waterproof protein, and that's why your skin has so much of it, and your hair and your nails. It's very tough, and it's very waterproof, and it helps to maintain homeostasis for your skin. So there's just so many different proteins, we can't go through them all. Now, this is the last concept, though, that you will be tested on. And this is the reason why I will explain it's not good to let your fever go up to about 105, 106 if you have a fever. Your body artificially resets your internal thermostat when you have a fever because a slightly higher temperature increases metabolism, and that helps your body to be able to fight off the infection. That's why you get a fever. However, it shuts off your ability to sweat, and as your body temperature rises, when you get to a certain point, there starts to become an issue. Proteins will only remain stable at certain temperatures. If the temperature starts to go higher and higher, due to the heat, the proteins will actually start shaking and disassembling in that tertiary structure. Remember, hydrogen bonds are weak. And so what happens is, if you raise the temperature enough, then the proteins do this. They essentially unravel. Now, what they don't do is this. They don't snap back together. Okay? It's not like a rubber band. If they unravel, the cell has to chew them up through hydrolysis and remake them all over again. When your cells get too hot, like when you have a fever, your proteins undergo this process. We call it denaturing. So you're going to need to know what denaturing is, as well as what can cause the denaturing of your proteins. So denaturing is essentially a loss of its quaternary or tertiary structure. It goes back to this. It just unfolds to its primary structure. It's like melting down a wrench or screwdriver. You have this molten, metal that doesn't do anything because it doesn't have any shape or any structure. Okay? That's why you don't want your temperature to get too high, because when the proteins denature, the cells can't do their job, the cells die, and then you start having massive issues from there. Now, temperature isn't the only thing that can actually denature your proteins. That's one thing that can disrupt the hydrogen bonds. Guess what? Since proteins are also held together by some ionic bonding as well as hydrogen bonding, if you change the salt concentration, sodium, chloride, potassium, calcium, any number of different ions, if you increase it or too much, then it disrupts the hydrogen bonds and the protein falls apart. So proteins in too high of a salt solution will cause them to denature. They'll break apart. So it's not just about heat. And the last one is pH. Remember, all pHs is a measure of the hydrogen ion concentration. Well, proteins are designed to work at very specific pHs. Not all proteins work at a neutral pH. Some can work in very acidic environments, like a pH of 2.7. Some work in more basic environments, like a pH of 8 or 9. Each protein is specifically designed to work at a specific pH. So if you change that pH by adding hydrogen ions or adding hydroxide ions, the same principle applies as it does with salt. The ions, the hydrogen and the hydroxide, will disrupt the hydrogen bonding of the proteins and cause them to denature. So, changes in pH, changes in salt concentration, and changes in temperature are pretty much heating things up. If you cool things down, you can stop the proteins from moving, but that's how we preserve proteins for long periods of time. They don't denature by freezing them, but they do denature by heating them up. So, I will test you on what things can actually cause denaturation. So what things don't denature your proteins? Well, let me give you a prime example that you'll probably see on your quiz. Light. Light doesn't denature proteins. In fact, it protects us. We have a defense mechanism. When you go outside and you're exposed to not only sunlight but UV light, your body reacts by producing a protein that absorbs the light called melanin. Melanin production. So melanin is produced by your melanocytes in your skin. That's why your skin gets darker. It's a defense mechanism. Why? Because UV radiation can damage your DNA. And so your cells produce this protein, melanin, to absorb the light. And they're not denatured by that. So that's just an example of something you would look for on this one, where I say which of the following would not cause your proteins to denature. One might be freezing them. That doesn't cause them to denature. Or exposing them to UV light or even just regular light doesn't cause your proteins to denature. In fact, we are kind of light driven. Vitamin D production, we need light for that to occur. So we do need some sunlight to be able to remain healthy and maintain homeostasis. But UV light is damaging. That's why your body reacts to it by producing the melanin. Now, last organic group, nucleic acids. Here we are coming back to where I mentioned that there are some sugars, which are technically carbohydrates, but they don't form the building blocks of the carbohydrate group, which is why we don't include them into the monosaccharides. Two of those sugars are called deoxyribose and ribose. And that's where DNA and RNA get their names. DNA literally stands for deoxyribose nucleic acid, and RNA stands for ribose nucleic acid. So the names are derived from the sugar that makes up their monomers. Now, the sugar isn't the only thing that makes up their monomers. Here is the basic building blocks of all DNA and RNA molecules. There is a phosphate group, a sugar, which will differ between DNA and RNA. DNA uses a sugar called deoxyribose. RNA uses a sugar called ribose. And then this is the key part right here. The nitrogenous base. This part of the monomer is what provides the information. Okay, your DNA and RNA are pretty much just blueprint molecules. They're blueprints for how your cells need to make the proteins to be able to function. So we'll explain in a second how that blueprint works. But this monomer, we have a specific name for it. It's called a nucleotide. So make sure you understand that that is the monomer of the nucleic acids group. It's called a nucleotide. So what makes up a nucleotide? A phosphate group, a sugar, and what we call a nitrogenous base. Now there are many different types of nitrogenous bases. For DNA, there's 4, and RNA, there's 4. And they share most of them in common, though there are some subtle differences. DNA uses, now the reason why we call them nitrogenous bases is because they've got a lot of nitrogen in them, and carbon, and hydrogen, and whatnot. So DNA uses 4 bases called guanine, cytosine, thymine, and adenine. RNA, on the other hand, doesn't use thymine. It uses guanine, cytosine, adenine, and it uses uracil. Now, this is just a precursor. You're not going to have to memorize these yet, but in lecture 10 you will. So you might as well learn them now. But DNA and RNA are almost exactly the same. They just have one base that they don't share in common. DNA exclusively uses this base. RNA exclusively uses this base, but the other 3 they both use as far as their nucleotides. So if the nucleotides are the monomers, what are the polymers? Well, by the nature of saying DNA and RNA, those are the polymers. What do I mean? The shortest strand of DNA in any one of your cells is 33 million nucleotides long. Yes, million. They're one of the longest polymers in your body. And some of your chromosomes have as much as 100 million nucleotides to them. So if you count up all of the nucleotides in a single cell, you have about 5 billion nucleotides for every cell. Okay? So this is by far one of the more important organic molecules because of the data that it holds, the blueprint that it holds. Now, there are some fundamental differences between DNA and RNA in their structure. Let's look at those. DNA is your genetic hereditary material. This is one aspect that I'll test you on for DNA. It is what gets passed on from one generation to the next. You have a baby. How do you make that baby? Well, most of you know how to make that baby. But you're fusing the DNA between 2 individuals. So men will pass on half of the DNA through their sperm. Women will have the other half of the DNA in their oocyte or the egg. When that fuses together, that's the genetic material that you pass on. This is about reproduction. The RNA is a messenger molecule. It tells the cell, it gives the cell the information that's contained on the DNA. So you'll understand more about this in the next lecture when we talk about cell biology and where DNA and RNA are found and whatnot. But DNA is kind of like having this huge cookbook with all the recipes in it. Well, when you're making your brownies, so to speak, you don't want your kids to start messing up your original recipe. So what do? You make a copy of that page. You don't care if that gets exposed to the flour and the sugar and the markers and all the good stuff that your kids are doing when you're baking your brownies. That's really the main difference here. DNA is the hereditary material. RNA is just a copy of portions of your DNA to tell the cell what to do. Notice that RNA is just a single strand. And DNA is actually this double helix structure, and this provides greater stability for this to be a long-term information storage molecule. Notice how it's held together in the middle. What do you think is holding the 2 strands together? Hydrogen bonding. Okay? So individually, again, hydrogen bonding is weak, but collectively, it's what holds the strands of your DNA together. Now, how are these assembled together? The same process we've talked about, dehydration synthesis. So here's one nucleotide, phosphate sugar and a base. It then is covalently bonded right here to the next nucleotide, phosphate sugar base. Covalently bonded here to the next nucleotide, phosphate sugar base, all the way down the line. So the backbone of DNA and RNA is actually where the covalent bonding occurs for dehydration synthesis. The middle of the DNA is actually just held together weakly by hydrogen bonds. Now, why is that important? Because in order to copy DNA, it's like opening up a book. It has to be able to be opened and accessed very easily. As such, the DNA has to be separated and then put back together very easily, which is why it needs hydrogen bonding instead of covalent bonding. Because the DNA is constantly being opened and closed, opened and closed. Now, there's one more molecule in the nucleic acids group besides DNA and RNA, and it doesn't play its role in information. It plays its role in metabolism. What am I talking about? It's called ATP, adenosine triphosphate. Why is this a nucleotide? Well, let's look at it. It's made up of a phosphate, a sugar, and a nitrogenous base, a nucleotide. So that's why it belongs to the nucleic acid group. Now, later on, we'll discuss all the ins and outs of this. For now, just accept that this is the battery that fuels all metabolism. When your cells need energy, this is what they tap into. You're like, well, why isn't it glucose? Glucose gets turned into this. Lipids get turned into this. Proteins get turned into this. So we'll discuss in lecture 7 how that's done. But that's really what ATP is. It's that AA battery, or that AAA battery, or whatever battery, that plugs into every metabolic process in your cell. So that's why this is also an important molecule to understand. It is a nucleic acid, or a nucleotide, because its fundamental structure is a phosphate, sugar, or base. We'll talk more later about what those other phosphates are and why. So now you know ATP, DNA, and RNA, these are all nucleic acids. Now ATP is a monomer. It is not a polymer. It's just a single unit. So ATP is an example of a monomer of the nucleic acid groups, where DNA and RNA are the examples of the polymers. Now, RNA isn't usually as long as DNA. RNAs range anywhere from several hundred to several thousand nucleotides. So they're still pretty long. They're not millions of nucleotides long like the DNA, but several thousand is still pretty long. So RNA and DNA are the polymers of that group.